Okay, so we still are just the participants are coming and they are in the waiting room. So maybe since we have already the two, I have to take off the sound of my. Okay, they are coming already.
Yeah, I think we are live, Cardinal.
Yes, we have 30 persons. I think Tom is adding. I thought it was me, but I had Blanca.
Hi, Blanca.
Yes.
Hi, Lisa. It's so nice to see you.
Nice to see you too.
Hi, Blanca. Hola, ¿qué tal?
Hi. Hola. It's a very good idea, this meet the expert thing.
Yes.
Congratulations for the idea.
Karina, we have around 14, 15 people on YouTube. So
Okay.
just for you to know.
Thank you, Vanessa.
Because some people can watch by YouTube.
Yes, I have the two computers, one with Zoom, the other one with YouTube.
Oh, there are people also from Florence. I've seen some names that I know.
Colleagues.
Colleagues.
Okay, we have 33. Okay, Tommy saying, hey all, please mute yourself when you join.
Hi, Amaya.
Hi, Amaya.
Amaya, hi.
Hola, Maya.
Hola, hola.
What do you think that we will start in 2 minutes?
You are muted, Gordon.
Sorry. I think that you can start. It's 3 minutes past 2 and we are going live.
Okay, so starting now. Okay, the chair is saying, okay. So, hi everyone. Hope you are safe and well. For those who don't know me, my name is Karina Ladeira. I am the Young Investigator Representative of the International Comet Assay Working Group. That is a specialist group of the European Environmental Mutagenesis Genomics Society. And we decided to organize two Meet the Experts this year in order to dynamize the dialogue mainly between young researchers who use or are willing, interested to use Comet Assay. I will just say for the new persons, can mute their microphones, please. Sara Ristori, if you can mute your microphone, please. Okay, so continue. We have this Meet the Expert with the purpose to dynamize the dialogue between young researchers and that are interested to use Comet Assay and experts that are using Comet Assay for long years. So my first message here is that I hope you enjoy this event. And now I want to give some information before we start. The meeting will be recorded, shared, and streamed in YouTube, so you can revisit as much as you want. And if you have any problem for recording, please do not put your face here. If you explore our website, you can see other videos that we have there. There are more technical videos of the Comet Assay technique. They are very interesting and dynamic. So I advise you to see, to explore that. And we have that Google Forms that you can drop your questions, but also you can still drop your questions here in the Zoom chat. Please write to everyone in order to the experts and myself to see your questions and have the chance of reading it. And also you can raise your hand and ask directly. Do not forget, if you do not want to be recorded, put your camera switch out when you raise the hand, if that's your objective. You can also write questions on YouTube. There is a delay of 15-20 minutes of the Zoom meeting with the streaming, but I will do my best for read all of your questions. So now let's start with our first expert, Professor Lisa Giovanelli. Professor Lisa Giovanelli, that is Associate Professor of Pharmacology and Toxicology of Florence University. She studied biological science in the same institution, and also her PhD was there. And her main research topics are studies on the damage as a biomarker of effects of environmental pollutants, degenerative pathologies, and aging experimental models in humans, and also studies on protective effects of natural antioxidant compounds towards age-related dysfunction and age-related markers in human, in laboratory animals, and in cell cultures. Also, she has been involved in many EU projects, and it was in that one of the projects, the Comet project, that we are all now together and we are doing these initiatives. So, Professor Lisa,
Yes,
I'm very grateful for you to accept this invitation and this challenge. I will ask you if you can share your presentation. Professor Lisa will make some presentation to all of us. In the end, you are invited to do your questions. It's perfect. And when you want to start, please feel free.
Yeah, I would like to enlarge the screen if I can. Yeah, can you see my screen correctly?
Yes.
So Karina, thank you very much for the presentation. Good morning to everyone, or good afternoon, depending on where you are. The title of my brief presentation is here, you know it, Blood Analysis with the Comet Assay, Advantages and Drawbacks of Different Preparations. And I will focus on 3 blood preparations that have been widely used among Comet Assay users: whole blood, Buffy coats, and peripheral mononuclear cells. I thought I would keep this presentation very simple and basic, considering that I don't know who is in the audience. Maybe there are also beginners. So I will be very basic, and then with the question, maybe we can go into detail of what you want. So whole blood analysis has been, of course, very important in science, particularly in human studies, because blood is easily obtained from humans, both patients and healthy volunteers, and also because blood cells are circulating all over the body, they can be seen as sort of sentinels of what happens in the whole organism. So DNA damage is one of the many, many biomarkers that we can measure in blood, and it can function as a biomarker of effect or as a risk biomarker, as also in the Comet European project has been demonstrated. So in recent years, whole blood has been increasingly used in human studies focused on DNA damage, both in studies on environmental pollutants and also in intervention studies. Of course, doing the Comet assay in whole blood means, as a matter of fact, measuring DNA damage in leukocytes, the only cells that have a nucleus in blood. So basically the mononuclear cells, lymphocytes or monocytes, and the polymorphonuclear cells, the granulocytes, mainly neutrophils. What are the advantages of measuring DNA damage in blood using whole blood preparations? There are many advantages that have been put in evidence in the last years, and that's why whole blood preparations are increasingly popular in this type of studies. First of all, using whole blood takes, of course, substantially less time compared with the isolation of cells, particularly of mononuclear cells. And this is an advantage for human studies in general and biomonitoring studies, for example, especially where in the case that you have large sets of samples that need to be processed all at once or in a short time. Also, only a few microliters are sufficient. And so this is very useful, either because you could have ethical problems in obtaining large amount of blood, or you might need blood for many other analyses, or you might want to stock different aliquots of your sample, and you need space for that. Also, avoiding cell isolation means to avoid potential induction of DNA damage during the isolation procedure. This can happen. And finally, blood can be easily cryopreserved, also without any cryoprotectant if you use very small volumes. The cryoprotection issue will be discussed in the next presentation, as you know, so I will not go into any detail on this, but probably we will have the chance to go back to this point also in the following presentation. On the other hand, there is another preparation from blood that has been and still is very popular, which is peripheral blood mononuclear cells. In the past, they were called lymphocytes, but they are actually mostly lymphocytes, but there is a substantial percentage also of monocytes in this preparation. So speaking about mononuclear cells is definitely more correct. What is the advantage of working on such a population instead of using the total leukocytes? Of course, this is a more homogeneous population, and lymphocytes are longer lived compared to the other blood cells, so they can work better when we want to see the effect of long-term exposures. So there are advantages in using this kind of preparation. We will see this also in the following slides, and that's why I think this preparation has been much used in the past years, much more than whole blood, I would say, especially in intervention studies. The preparation is rather well shared among laboratories, meaning that there are not so many differences in the preparation going to one report to another. Basically, diluted blood is stratified over a dense mixture, which can be lymphoprep or FICOL or other type of preparations, and then a density gradient centrifugation follows at the speed of 1000 Gs to obtain, in the end, as you can see here in the picture, a layer where you have an enrichment of peripheral blood mononuclear cells. You can then aspirate this and then resuspend it and use it for directly the Comet assay, unless you want to manipulate the cells, treat the cells in some way before analyzing DNA damage. And there's a third preparation that I want to talk about, which is Buffy coat. Buffy coat has not been used so much to measure DNA damage in human studies. It is not so popular, let's say, so much used as the other two preparations that I talked about. But Buffy coat is very interesting, and we have investigated the possibility of using it also in the Comet project. In general, this is used to prepare human donor leukocytes and platelets. So huge amounts of Buffy coat can be prepared, for example, in the hospitals. It is used to prepare DNA from blood, to differentiate leukocytes in a smear, and much important for us DNA damage researchers, it is stored in biobanks. Biobanks use Buffy coat samples for long-term storage, even more than whole blood samples. So the possibility to use Buffy coat samples from biobanks is, of course, very important, because it means the possibility to engage in very large prospective studies, possibly multi-center studies. And once again, I think that Helga and Eilul will talk about this, because they will talk about the cryopreservation and of course, if we talk about biobanks, we are talking about cryopreserved material. So what is Buffy coat? It is the top layer obtained after a low-speed centrifugation of anticoagulated blood, as you can see here. It contains most of the leukocytes and platelets. So once again, it's a leukocyte preparation. But it is not pure leukocytes, because it contains a lot of erythrocytes. Basically, it is a sort of concentrated preparation of leukocytes, meaning that compared to whole blood, they are more concentrated in comparison to erythrocytes, which are also present anyway. The preparation of Buffy coat is not so consistent among laboratories as it is the PBMC preparation. If you look at the literature, you find that this low-speed centrifugation can actually go from 200 to 1,000 Gs. And we have seen that this can make a difference, both in terms of the number of cells that you recover and in the proportion of different leukocytes, of leukocyte subtypes in the Buffy coat. Not only that, but also in the literature, you find sometimes that authors refer to Buffy coat for gradient isolated leukocytes. So let's say that for Buffy coat, the situation is less clear for the preparation compared to what it is for the peripheral blood mononuclear cells. A few years ago, we have done in Florence some experiments with Carina Ladeira and my collaborators, and my collaborator Francesca Scavone, with the specific purpose to compare from the point of view of the measurement of DNA damage with the Comet assay, these three blood preparations. Here you have some examples of the Comet images that you can obtain from the three different preparations. You see that when you do the Comet assay on whole blood, here you have control undamaged cells, and here you have a Comet. You always, in most of the cases, you get these sort of ghost-like round images around each nucleus, which we suppose are remains from the erythrocytes. Erythrocytes, of course, are much, much more numerous than the leukocytes in these preparations. But we have noticed during the image analysis that this is not a problem for the image analysis itself. In the Buffy coat, you also can obtain this phenomenon because you still have erythrocytes around. It might be a little less pronounced, but you still find this type of images. And also in these preparations where you have leukocytes, so not only mononuclear, but also polymorphonuclear, you can see in some cases polymorphic nuclei, like this one, for example. So you can distinguish to a certain extent the mononuclear from the polymorphonuclear cells in these preparations. When you analyze the PBMC or PBMNC, as we called them here, the situation is different. It is clear that this is a much cleaner preparation. You can see it from the images. So the controls are totally round. There is no ghost-like image around, and the same thing is true for comets. So we measured the levels of DNA damage in these three preparations, and once again, I will focus on the fresh samples in my presentation. And here you can see the results of the measurement of DNA damage with the Comet assay expressed as percentage DNA in whole blood, Buffy coat, and peripheral blood mononuclear cells. We measured strand breaks, and these are the darkest columns. FPG sites, which is oxidative DNA damage on purine bases, and these are the intermediate gray columns, and also DNA damage exogenously induced by hydrogen peroxide, and these are the light gray columns. Let's start from the basal level of DNA damage, strand breaks and FPG sites, oxidative DNA damage. As you can see, there is basically no difference among the three blood preparations. Very low levels of DNA strand breaks and rather low level, very similar in the three preparation, also for oxidative DNA damage. On the other hand, we can see a huge difference in the response to hydrogen peroxide of these three different cell preparations. The fact that whole blood does not respond to hydrogen peroxide is widely described in the literature. We were not expecting nothing different, and indeed this was the case. Why is that? Probably considering that we have done, as most of the people do, exposure to hydrogen peroxide when the cells were already embedded in the gels before the lysis, of course. Probably the point is that in the gel you have a lot of erythrocytes, a lot of plasma proteins that exert a sort of physical protection on the leukocytes from hydrogen peroxide and free radicals originating from hydrogen peroxide. And also there is a lot of antioxidants in this preparation, both in plasma and within the erythrocytes. So probably we think this is the reason why, if you try to challenge whole blood preparations with hydrogen peroxide, you basically don't see an increase in DNA damage in the leukocyte that you are measuring for DNA damage. In Buffy coats, we observed a slight increase in the effect of hydrogen peroxide. See, we used a very high concentration, 500 micromolar, compared to whole blood, but still not so, not such a big increase. And that's probably why, as I was saying before, erythrocytes are still present. They might be less, but they are still present. Whereas in PBMC, as you can see, the effect of hydrogen peroxide was very important. There is a huge response in terms of increase in DNA strand breaks in these cells. Now, why is this type of ex vivo stimulation important? Well, particularly in intervention studies where many times you are working on healthy subjects whose basal levels of DNA damage in their blood cells are very low. If you only measure the basal levels of DNA damage, it is very difficult to imagine that you can increase, that you can further reduce these levels. So that's why many intervention studies also use challenging the cells, the samples with hydrogen peroxide to basically assay the antioxidant ability of the cells. For example, if you are doing an intervention with an antioxidant, with a plant antioxidant, for example, or a food which is supposed to have antioxidant properties, you have a good probability to see an increase in antioxidant capacity of the cells, which is a reduction in the effect of hydrogen peroxide measured in this context. Here you have an image of one of our Buffy coat preparation. You can see very well many leukocytes, both polymorphonuclear and mononuclear, and you also have erythrocytes around. What I wanted to show you in this graph is DNA damage in a Buffy coat sample upon induction with hydrogen peroxide, plotted in a different way, expressed as frequency of DNA damage. That is, you plot the frequency of the cells against the intensity of DNA damage. And if you do this, you can observe that, yes, there is a number of cells, about 40%, which exhibit intermediate or high levels of DNA damage upon hydrogen peroxide challenge. But there is a high proportion of cells, about 60%, that, as a matter of fact, does not respond to hydrogen peroxide because the induced DNA damage is lower than 10% percentile DNA. So once again, the preparations of Buffy coat might definitely be very much useful to measure the basal level of DNA damage in human samples, but they do not seem to be so useful when you want to assess the antioxidant capacity of cells, the variations in antioxidant capacity with exogenous oxidative stress. Of course, these graphs always tell us something about the variability within our samples. All the 100 cells or 200 cells that we measure within one sample in the Comet assay, they are not exactly the same. So there is some variability. The same can be seen also in peripheral blood mononuclear cells, although this is a much more homogeneous sample. However, you can see when you look at the frequency of hydrogen peroxide-induced damage that there is some variation in the behavior of the cells that we have measured. However, in this sample, as you can see, most of the cells definitely respond to hydrogen peroxide with a high increase in DNA strand breaks. And there is only a very small percentage of these cells that do not respond at all. On the other hand, we have to think that also the peripheral blood mononuclear cells are not so completely homogeneous, of course. There are monocytes, there are B and T lymphocytes, and there are the different subtypes of lymphocytes that presumably can respond to hydrogen peroxide with different sensitivities. So this is another thing that, of course, we have to keep in mind. So I will, at this point, summarize this brief and hopefully clear presentation. The bullet points are: whole blood is, I would say, definitely the most straightforward preparation from many points of view, as I have tried to describe. Fast preparation, small volumes, easy cryopreservation. However, I have shown you that preparations where plasma and plasma proteins and erythrocytes are present do not allow the use of exogenous oxidative stimuli to evaluate the antioxidant response ability of your cells. So they are basically useful to measure the variations in the basal level of DNA damage. And you can do strand breaks, FPG sites, endo3 sites, whatever you want. But in all cases where it is very important to find interesting results, to measure the antioxidant response of the cell, I would say the best preparation is still probably the oldest one in this research field, the peripheral blood mononuclear cells. And this is all I wanted to tell you as a beginning of the discussion or the questions that you will want to pose. So thank you very much, you all, for your attention.
Thank you, Lisa, for your very good presentation. There was already a question in the chat, but
Excuse me, Karina, shall I stop the...
Yes, if someone have a particular question about some of the graph or something, we can go again, but now it's more nice. We have a question in the chat regarding the peroxide oxygen treatment that we did, and since I participate also in the study, I already answered, but I would like you to confirm, because I have the paper with me and I checked that we used 5 minutes at 4 Celsius degrees.
Exactly. We use 5 minutes. It is important that we use the 4 degrees because in that case you avoid all the antioxidant enzymes. Otherwise, you would probably need more time. I can see that Goran for example, he uses 10 minutes. Of course, there are some variations in this protocol.
I remember because we use 500 microliters that at the time we were asked by the Comet why we use 500 and not 100. I don't know if Gordon used 100. That's why he used the 10 minutes. I don't know. I don't know if you want to say something.
I was using a little bit less concentration, so because of that I use a longer period of time, but same as you on 4 period degrees. I also have a question and wanted to comment. When I also use hydrogen peroxide with whole blood in gels, sometimes I have this population of cells that is really highly damaged and a population of cells totally intact. So I see it more like this. Then I get like this background value of DNA damage that is higher than compared to control, but I always have population of cells that is totally normal and then with the huge tails.
Okay, we have another question here. Well, so thank you very much for the event invitation. So it's from Turkey.
And in the team of Nursen.
Okay, yes, our colleague and friend. So the question is, during exposure, the iron in whole blood is likely to cause DNA damage as a result of the Fenton reaction. However, it seems contradictory that H2O2 exposure does not cause DNA damage. Could the reaction with iron from hemoglobin in whole blood be responsible for the reason why no damage was observed in H2O2 exposure for whole blood? So it is a question that we had discussed already.
Yeah, I think that the problem with hemoglobin iron could become important upon lysis of the erythrocytes. So, for example, there are many preparations of leukocytes that involve erythrocyte lysis, and in that case, I think you have to be very careful because all the iron could evoke oxidative damage on leukocytes. But when we do hydrogen peroxide stimulation, as I described, as we were talking about also with Goran, we are before the lysis step. So the erythrocytes are not lysed. And so we are not dealing with that particular problem. In the next step, when you do the lysis in such a high volume compared to the volume of your gels, that even if your erythrocytes release iron, that's so much diluted that apparently it doesn't have the time to do any damage, because when we go back and look at our results, as you can see from the ones that I have shown, but of course, these type of studies have been done by many other people before us, you don't find high levels of DNA damage in your leukocytes, meaning that the problem of the release of iron during the lysis step by erythrocytes is not a real problem. Was I clear? Did I answer your question? Good.
I was searching in YouTube if we have any question there. We do not have. I don't know if someone want to share questions, but I can also ask some question to Professor Lisa, because this is intended to be for young investigators. Of course, all of us still have questions. For the ones that are starting and have the possibility to design a study, which type of sample would you advise according to your presentation?
Yeah, well, depending on the type of study that one wants to do, I would suggest to use peripheral blood mononuclear cells if we are doing an intervention where we would expect to see lowering of DNA damage levels. Because once again, if these are not patients, of course, if you start with patients with a high level of DNA damage, like for example, diabetics, then you can use also whole blood and see whether your intervention has been able to reduce DNA damage. But if the subjects are normal subjects, healthy subjects, the basal level of DNA damage, as I showed, because those were healthy subjects, maybe I didn't say that, but that was the situation. In that case, the basal level are very low. And so you need to be able to see the antioxidant effect in another way. In that case, you need hydrogen peroxide, and so you need peripheral blood mononuclear cells. And again, and also when you do observational studies, for example, you want to see the effect of pollutants or other type of environmental toxins or toxic substances, then I think that probably, because in this case you are expecting an increase in DNA damage, probably whole blood could be a good solution for you, especially if you are expecting to do large numbers of samples. If the samples will not come to you early in the morning, but maybe they will arrive in the afternoon or in the evening, then you do not have time to run the Comet assay. We also have the cryopreservation issue, and Helga and Elul will tell about, but using fresh blood could be a good alternative in this case. In some other situation, you might be so lucky that you can do both, whole blood for certain things, and also prepare peripheral blood mononuclear cells to have a more precise, homogeneous population to obtain answers on the long-term exposures. So it really depends on the type of study, the number of subjects, and the experimental situation that you are facing.
Yes, for sure. You said now a very important thing, that it was, again, another question that I would ask is, because we have this case of peripheral blood mononuclear cells. They are more homogeneous group, and they are a long-lived population compared with leukocytes. So they can give us a more complete information about the cumulative exposure. However, Comet assay is a type of biomarker of immediate effect that can be repaired or not. So sometimes I have this philosophical question, because if it's the immediate effect that could be repaired or not, should we search for these mononuclear cells, or can be just leukocytes? In case of micronucleus, I understand, because there is more cumulative exposure. So sometimes I have these issues to try to understand, but of course it depends on the type of the study and other biomarkers that we are studying that are involved. Because for instance, if someone will run micronucleus assay, in the end, they can cryopreserve small amount of sample and do the Comet assay in the mononuclear cells. It's a way of designing experiments. I think that we do not have more questions. And so, Lisa, I want to thank you once again. And
Thank you all.
personally say that I miss our investigations.
Me too, but there will be other chances,
Yes,
I'm sure.
Yes. Okay, so now.
Thank you all.
Thank you. Now we'll go for the next two experts. Professor Helga Stopper, that is Professor of Toxicology in the University of Würzburg in Germany. She studied biology in the University of Regensburg in Germany also, and she did her PhD in the University of Würzburg in Germany, the Institute of Biotechnology. After that, Professor Helga joined the Institute of Pharmacology and Toxicology in the same university as a postdoctoral scientist and later as group leader. And then she became Professor of Toxicology, and she was acting chair between 2009 and 2019. She also was president of the German-speaking local society of GUM and president of the EEMJS from 2017 to 2019. I want to say my thanks also for Professor Helga Stopper for promptly accept this invitation and challenge to be here with us.
Thank you very much, Karina, for the nice introduction, and thank you for the invitation. Shall I share my screen now?
Yes, please.
Okay, we can see it. Okay, great. I'm going to give a short presentation about freezing of cells for genotoxicity and DNA repair studies, and then Elul Bankulu will give another short presentation afterwards about the use for a study that we did. My presentation will be more general about freezing and thawing of cells. So there's a general protocol for freezing of cells that we all know, and it's also true for freezing of lymphocytes, where we isolate lymphocytes using a gradient density centrifugation. We wash the cells, we adjust to about 1 × 10⁶ per milliliter. We use a cryoprotectant. We then usually use elevated serum concentrations of about 10 to 20%. And then we put the cells in a slow freezing container, which is either an isopropanol filled box that we put at -80 degrees, or it's a specific equipment which cools down the cells to about 1 degree Celsius per minute. If you want to store the cells for a longer time, then we usually transfer them to liquid nitrogen and store them in the gas phase above the liquid nitrogen. Now, the question is this the right way to do it? Are there alternatives? And how long can you keep the frozen cells like this? And probably most publications agree on this. Probably you can store them in liquid nitrogen for decades, maybe even centuries. At -80 degrees, it's a different question. At -80 degrees, it probably depends on the conditions, and it also depends on whether you need all of the cells alive after thawing, or maybe just 80% or 90% or 70%. For normal cell culture, it's usually enough if we have, I don't know, 70% of cells alive. We culture them, we wash away the dead cells soon after, and we have a fine culture. But there might be different approaches where you need almost all of the cells alive. I want to share with you this quote that I found on the internet, and the source is shown here below. And I think that's very important and very typical for this issue of whether we can keep cells in a regular -80 degree freezer or whether we have to store them in liquid nitrogen. And I want to read this to you. And this is from a researcher who said, I recovered a couple dozen drug-resistant cancer cell lines from the same -60 degrees freezer. All were viable between 10% and 70% after 8 to 10 years of storage. None were ever at a temperature lower than -60 degrees. One important point though, the -60 freezer was very well organized and cataloged and was for cell storage only. Access was very limited and no one was allowed to spend more than a minute retrieving cells. You were watched by the person in charge of the freezer, so as to keep the temperature constant at -60. And the same person says, today, next to my office, there's a -80 freezer where grad students stand in front of the open door, casually going through disorganized boxes, looking for a vial of frozen Buffy that doesn't even need to be there at -80 degrees in the first place, and letting the temperature go as high as -40 on a daily basis. I hear the alarm at least half a dozen times a day. Such a freezer would clearly be inappropriate for cell storage. On the other hand, I think that probably more than 90% of what is being stored in liquid nitrogen in most labs nowadays doesn't need to be there. I think that's a very important point when we think about, do we have to store our cells in liquid nitrogen or can we store them in the -80 freezer? I think often we can store them in the -80 freezer, but we have to make sure it's actually really -80 degree. It does not go below, because what we often don't consider is that even at temperatures of -40 or 30, some little spots in the cells start thawing, and that could damage the cells. Usually, we hold up the vial and we think, okay, it's still frozen, that's fine. But that's not the case. We cannot rely on that. Inside of the cell, the little tiny areas that are not frozen anymore can damage the cell. Okay, if you really do keep them in -80 degrees consistently, they can be kept for a long time. And there's, for example, this paper showing it for human HepG2 cells and also for bovine carotid artery normal endothelial cells. They looked at the cells for 8 years, and the viability that they saw was over 90%. And it was the same as for those that were stored in the liquid nitrogen phase. And also the proliferation of the cells was fine and not different from liquid nitrogen. Recently, the question was brought up to me whether you can freeze cells without a cryoprotectant. And the answer is not really. But there's one paper which claims that they could do it. And this is a paper where they use a super flash freezing technique. And that is an ultra rapid cooling using inkjet printing. So that sounds like something that's not available to everyone. It's a very fast freezing method, and even usually when you do very fast freezing methods instead of the slow freezing variation, the fast freezing methods are sometimes used for stem cells, and it's sometimes called vitrification. And even for that, you usually need a cryoprotectant. But this group here claimed that they can do without, with this special equipment and method, and they looked at a cell line and at stem cells, and they found no difference in viability between conventional freezing and their method. And also the expression of stem cell markers was not different, or was still there for the cells that were frozen and thought was their method. So now what does that mean for the Comet assay? Now in the Comet assay, we have to remember that usually we don't need cells which are able to proliferate. We just need cells that still have a nucleus where the damage has not been changed due to the freezing and thawing procedure. So that's a different situation. And here we have an example of cells that were fresh, and from the same cell samples we have cryopreserved cells, and we had whole blood, Buffy code, and PBMCs. And you can see that there is no significant difference between the different blood preparations. Also in this particular study, PBMCs were a bit higher, but not significantly different. And there's really no difference in damage between the cryopreserved ones and the fresh ones. And what we see here is the basal damage in white color, and the gray bars, they show the additional damage by addition of FPG enzymes, so the additional oxidative damage. So we can use cryopreserved cells for that. Usually the PBMCs are prepared with cryoprotectant, and whole blood and Buffy code not. Okay, now the question of repair. Can we also do a DNA repair assay with the Comet assay with frozen samples? Now we like to use a certain variant of the DNA repair assay that I would like to briefly explain to you. There are other variations, but we use this one. And we expose the cells initially to hydrogen peroxide or UV radiation. Then the cells try to repair this. They cut out the lesion, and then they have to fill in the lesion by either base excision repair or nucleotide excision repair. This filling of the lesion involves DNA synthesis, and the enzymes that do that can be inhibited by acetylcholine. So that means in this assay, in this variant of the assay, the incision activity or the repair attempt of the cell is revealed by an accumulation of breaks because of the inhibition of acetylcholine. So this shows the scheme of the assay, and here we can see that, for example, with UV, UV alone, the left bars, doesn't do damage in a large concentration range, and also acetylcholine alone, here shown, does not cause any damage by itself. But if we have UV and then for a certain repair time, we add acetylcholine, then we get the damage. And this is due to repair attempts of the cell. Now, if you do this with peripheral blood lymphocytes, mononuclear cells, then it looks like this. We have a control, and this is after repair time, so not different. We have a control with acetylcholine after repair time, not different. We have a treatment with hydrogen peroxide, which causes the damage. And if we do not add acetylcholine after the 2 hours repair time, we already have a reduction of damage, of course. And if we induce hydrogen peroxide mediated DNA damage and we add acetylcholine, we get an increase of damage. And this is our repair-associated additional increase. Now we tried this with cryopreserved PBMCs, and here you can see that all the black bars, so all the samples after the repair duration, have a high DNA damage value. And here this looks like the one on the left side, but all the others are also high. So we cannot interpret this increase as DNA repair anymore because all the others have the same difference to their controls. So that means in the cryopreserved samples, this type of repair assay with freshly thawed cryopreserved PBMCs did not work. Now coming to thawing of cells. If one wants live proliferating cells, then you have to thaw the cells fast. For example, in a 37 degrees water bath, you have to wash off the DMSO by transferring to a large volume of pre-warmed medium. And with some very sensitive cell types, it's recommended that you add the cell solution dropwise to a pre-warmed solution to preserve viability even better. For the Comet assay, and in order not to induce any additional damage during the thawing, you also thaw the cells fast, but then you keep them on ice immediately after thawing, and you try to use them as soon as possible. And you have to be aware that the basal DNA damage may increase during the first hours after thawing, as I've just shown you in our example. And we think this may be due to a percentage of cells that are dying during these first few hours after thawing. So in these first few hours, we cannot do a repair study. We have seen that after an overnight recovery, the peripheral blood mononuclear cells may be used for an in vitro challenge with compounds or for a repair assay. But we do not know whether this is also true for cells that have been stored for a long term. There's still work needed to answer this. Now, freshly thawed lymphocytes, they may be used for a micronucleus analysis. And there has been some recent progress in this, and I will show you a slide soon. But there also you have to consider that a percentage of the cells will die after thawing, and you have to think whether this is acceptable for your planned study, because you can never exclude that the cell death is maybe somewhat selective. So this is an example of an older study from 1990, where they used 69 fibroblast cell lines, and they thawed them, and they compared the immediate micronucleus frequency to the micronucleus frequency after 3 subcultures. So that's always the right bar is after 3 subcultures, and the left is immediately after thawing in the first cell division. And from the 69 fibroblast lines, about 2/3 showed micronuclei in the normal range after thawing, but 1/3 showed elevated micronucleus frequencies like those here. But in all of them, the micronucleus frequencies returned to normal after subculturing for some time. They also checked with 2 newly established cultures, and they found the same. After establishing the cultures, micronucleus frequency was low. Then after freezing and thawing, it was high, and after some subcultures, it was low again. So we consider it normal for regular cell culture that if we thaw the cells, we have to culture them for for a few doubling times before using the cell culture. But sometimes we don't remember this in the context of lymphocyte work, where we want to prepare the samples from a study with, with human study, population study. So I promised you this paper where a group recently has published what it seems good method for analyzing micronuclei from samples from cryopreserved blood samples, and they used whole blood, and they did micronucleus assay immediately after thawing. They checked with radiation induction of damage whether the micronuclei and the nuclear division index was okay, and it was very good, very similar. And they also repeated the analysis after several times after freezing. So they were stored for up to a year in the freezer, and they thawed them, and the results were fine. And I'll show you in the next and then last slide, I will show you data. But I want to show you the procedure first here. What they did is that they added whole blood dropwise to an equal volume of cold cryopreservation medium, which contained 80% field half serum and 20% DMSO. The samples were frozen in 2 milliliter cryo vials, and each aliquot consisted of 1 milliliter whole blood and 1 milliliter cryopreservation medium. So in the end, they had 40% serum and 10% DMSO in there. They tried different variations, and this is the one that turned out to be best. And then they placed their cryo vials in a Mr. Frosty freezing container, which is a isopropanol filled box, and put them overnight at -80 degrees. And then they transferred the samples for long-term storage to liquid nitrogen, where they had them for 1 week and up to 1 year. And here's the data from this study. And we can see different radiation doses. We can see a micronucleus frequency here, and this is always fresh blood on the left side and cryopreserved samples on the right side. And it is a very good correlation between the fresh and the cryopreserved ones. And also the nuclear division index, so the proliferation capacity of the thawed samples on the right side, is as good as almost as good, at least, as from the fresh samples on the left side. So this is all I wanted to say for now, and Elul is going to show some data which is linked to my presentation pretty much. And so I think I'll stop sharing my presentation so that Elul can continue.
Thank you so much, Professor Elga. Since the team is with Eyul also, Dr. Eyul, I will ask for Dr. Eyul share her screen. I will introduce her, and then in the end, the questions regarding this theme can be asked for both of our experts. So before Eyul start, I want to thank you also to Dr. Eyul for being here, for accepting this challenge. And I can say that she's a postdoctoral researcher in the working group of Professor Elga Stopper at the Institute of Pharmacology and Toxicology, University of Würzburg in Germany. She studied food engineering in Turkey, in Izmir, and then she did a master in food engineering also. Then she joined the working with Professor Elga in the Institute of Pharmacology and Toxicology when she continued to do research. And the main research interests is genetic toxicology in association with chronic diseases, with emphasis on obesity and metabolic syndrome. And also, Dr. Eyul Bankoklu is a European registered toxicologist and our colleague in the Comet cost when we were together all until 2020 when cost action ends. So the screen is already shared, and you.
Yes, and thank you, Karina, for the invitation and the opportunity of sharing my results. I think you can hear me well and see my screen, or?
Yes.
Okay. I am going to add some more information about the usage and storage of human blood samples for analysis of DNA damage with the Comet assay. In most cases, actually, Can you see the next slide? Because I can't.
No.
Kind of. I think there is a delay. Sure.
Okay, okay. We are seeing already.
Yeah, okay. Okay, what I want to say, in most cases, the use of fresh samples is the method of choice, actually, especially to avoid the deleterious effect of freeze-thaw cycles. But there are some special cases that using cryopreserved samples are very useful and have some advantages. One of the examples for this might be working or collecting samples from various locations from several different countries that you need to use cryopreserved samples, or another example would be human biomonitoring studies. Then cryopreserved samples can offer you several advantages, like running samples in the same time, and you can also control and reduce the day-to-day variations by this way. However, they also bring some difficulties, which we need to be aware of and discuss and eventually try to control. One of them is increased background DNA damage due to freeze and thaw cycle. Another problem is possible effect of freezing medium, especially on enzyme activity. And in case of, as Professor Stopper already shared, if you like to culture cells after thawing, you might need to consider the dead cells and lost population, and due to this kind of selective population in this culture. And of course, culturing and storing cells long-term needs some facilities. So you need a good freezer that can keep the temperature safe. And in case of a freezer malfunction or shortage in electricity, you can lose all your samples. As you can follow so far that cryopreserved samples are especially important for human biomonitoring. And I would like to shortly mention that if the second slide will appear. I have somehow a delay here.
No problem, it will appear.
But you can't see the second slide, or?
No, until now, no.
Never had the problem, so first time.
I don't know if you want to.
Maybe I can stop for a short moment and try it again. Sorry for it.
No problem.
So in a weird way, it started from the second slide again.
Mm-hmm. We can, if you want to, okay.
Or I will stop presentation mode if it's going on like this, and try to do without presentation mode. So what I wanted to say, what is the importance of Comet assay actually in human monitoring? So Comet assay is one of the most common used method for measuring DNA damage, and DNA damage is an important biomarker linked to human health. And especially primarily when we consider DNA damage, it's associated with cancer. However, manifestation of cancer from DNA damage take decades long. That means we need long-term observational studies. So there are several case control studies with exposure scenarios, which requires much shorter time. However, there are not enough retrospective studies to establish the predictive power of Comet assay and DNA damage as a biomarker. So basically, we need good controlled respect of the studies, which requires cryopreserved samples. There are two ways of doing it. We can start collecting samples. We need a long time, 30 years maybe, or we can just use biobank samples. That would be the best approach and shortest way, because they have also detailed medical health records from these individuals. However, there are some important factors because they require to work with cryopreserved samples which were stored long-term, and we have so far experience with shorter-term storage, but not decades long. So they bring some factors which need to clarify. First factor is, as you heard already from Lisa Giovanelli, that searching or picking up the correct material is very important. So it's the question what kind of cell or materials and suitable storage. And the second, that we need good established methodological controls to control the fluctuations and possibly observe the stability of DNA damage over time. And there are also some other factors can contribute our outcome that we need to understand, like the type of collecting samples or bench time or shipment, how these fluctuations during these times can affect the outcome. And last but most importantly, stability of DNA damage in these cryopreserved samples over time. So I will try to go through and show some of our pilot studies to explain these factors better. So first of all, we try to establish a methodological assay control by using a cell line, TK6 cells. We have either treated them with a solvent control or with a potassium bromide to especially induce FBG sensitive sites, and cryopreserve those samples and thawed them in different time points and run FBG Comet assay to observe the day-to-day variations as well as the effect of cryopreservation. As you can see here, we do not have an increase in the basal damage, and we can first see induction in FBG sensitive site via FBG incubation in the potassium bromide treated samples, and the standard deviations are quite in acceptable range. So we were able to establish a good positive control and negative control to run always with our human biomonitoring samples. After this, what we wanted to see effect of storage and transfer condition. There was also another background for this test, because we did know biobank samples were not available in huge amounts. They are limited amount of samples, and some were already thawed. So we wanted to basically understand, can we somehow deal with the thawed and refrozen samples, and what happens if the samples thawed during the shipment? So we got Buffy code samples in stores, we thawed them and directly performed Comet assay in the condition 1, and we kept them 2 hours on ice after thawing, here condition 2, and kept them 36 hours at 4 degrees after thawing, condition 3, and we freeze them, keep them -20, and thaw them again to perform Comet assay. As you see, except directly use after thawing, we have all other conditions elevated basal DNA damage. So basically, we need to take care of the cold chain. And if the samples are thawed, you cannot freeze and reuse them. It's not feasible for our aim. To explain the impact of cryopreservation better, I have here samples from 6 individuals that we have. They were cryopreserved PBMC samples, were either used as freshly, you can see in the black bar, or the gray color bar, cryopreserved, thawed, and keep at 37-degree culture for 1 hour, 2 hours, and up to 16 hours. You see the fresh samples doesn't show elevated damage, and frozen samples show no significant difference directly after thawing. However, within 1 and 2 hours culture, they have increase of DNA strand breaks, which is getting back to the control level again after 16 hours. Together with this, what we recognize, cell count was reduced to 25% compared to the initiation point after 16 hours. To explain this better, I have here a distribution of cryopreserved PBMCs in different DNA damage categories. If you see, after the fresh cells, after thawing directly at 0 hours and it's 16 hours, we have most of the cells in the damage categories from 5 to 30%, and less, almost no cells above 40% DNA damage. However, within 1 and 2 hours, we have cells appearing in the categories with more than 40% DNA damage. So we can say elevated DNA damage is here in the culture due to the small number of highly damaged cells, which are disappearing over 16 hours during the recovery. And they are exactly the number of cells which one slide prior, 25% of the cells, so disappeared cells. That means one need to really consider, when you need to culture the cells for repair activity or for treatment, that you need to consider the cell count and these disappearing cells and what would be the outcome, effect of these disappearing cells on your outcome. So after gaining some experience, what we did, we have run a pilot study by using the European samples from European Prospective Investigation in the Cancer and Nutrition in a shorter way, EPIC study. EPIC study is one of the largest cohort study in the world with more than 500,000 participants. And the samples are recruited across 10 European countries, followed almost 15 years. And the guess numbers for cancer cases by now about 100,000. So this is a perfect study to establish the predictive power of Comet assay. So we had a collaboration with the German Cancer Research Center in Heidelberg, and they supplied us some of the samples, and they are part of the EPIC study with more than 25,000 participants. So available samples were Buffy code and isolated PBMCs, but Buffy code samples were already collected at the beginning of recruitment, so at the baseline. And during the follow-up, they have also collected PBMCs, but they didn't have any PBMCs from the baseline. So we got samples at the baseline from 94 to 98. And from re-invitation, we received the PBMCs and Buffy codes almost 14 years apart. And 1 year apart that we had re-invitation time point 2 PBMC samples. You can see on this side Buffy code samples from 50 subjects. They have we have the x-axis, the baseline DNA damage from the Buffy code samples, and re-invitation time point 1 in the y-axis. And we observed only a weak correlation between the DNA damage over time. Here you have the PBMCs from 99 subjects, and we have the measured the DNA damage from re-invitation time point 1 and 1 year apart re-invitation time point 2, and we could observe a good correlation between time points. We have also conducted some further analysis by using these samples, but we have done, since we have an interest in obesity and effect of BMI on DNA damage, so we mix from our own collection cryopreserved PBMC samples into the EPIC samples in a coded manner, and we run them together. After decoding samples, we could observe that group of subjects with BMI values 30 and higher showed a significantly higher FPG sensitive sites. And here in this graphic, you can see that when we plot them together with BMI ranges from 18 to 70 together with DNA damage, we can see a linear relationship. So that would mean any amount of weight loss can help to reduce DNA damage, as well as any amount of excess body weight can contribute to DNA damage. However, we still didn't have enough data points between the 30 to 40 BMIs. That's why I do not want to conclude it 100% on this. But all this outcome showed us that cryopreserved samples actually are suitable for determining DNA damage. Long-term stored biobank samples also can be used in Comet assay. Especially DNA damage data from PBMCs show a good consistency over a time for an individuals. They are promising and suitable for such study, and Buffy code samples need to some further establishment and understanding for this aim. Although we are very positive about the long-term usage, long-term stored samples and usage in Comet assay, there are always some buts that one need to think and consider in such a study. For example, the bench time, composition of the freezing medium, storage conditions, they can all affect the outcome. And it's necessary 100% to have good methodological controls to know your variations over time. And it's necessary to control the stability of DNA damage with the help of the methodological controls over an extended time. And we would suggest, if you like to culture the cells after thawing, a concurrent cell count and viability test might be very helpful. So with this, I am at the end of my presentation. I would like to thank German Cancer Research Center, our partners, Dr. Kuhn and Professor Cox, for supplying such valuable samples, of course, and Comet Cost Action, creating such a great exchange and making all this information possible. And I would like to thank my colleagues that patiently run the several mini gels and, of course, scored the cells patiently months long. And thank you for your attention.
Thank you so much, Ayu, for your presentation. Now I would ask to, okay, so we can have some questions. So in the chat, I saw that any question is there. In the YouTube, also no. I don't know if someone want to raise his hand. I have some questions here, so maybe I can start. Questions for both of you. Blanca raised her hand, so I will, please.
Thank you very much for the three of you for these interesting presentations. I have a little doubt, maybe for Helga or for Ayu. How important do you think it is to store the samples using the Mr. Frosty thing? Because when you are running a human biomonitoring study, this may be a little difficult from the logistical point of view, because in many days you collect samples from, I don't know, but 20, 30, 40 individuals. So using these Mr. Frosties can be very difficult because you don't have enough devices and you don't have also enough space in the freezer to store the samples like that.
Maybe I can tell something about it and comment on it. So me personally, when I collected human samples, sometimes received 10 samples at the same time. You are happy to receive that amount of samples, but you want to freeze as much as possible. Then you recognize that all the Mr. Frosties are full. What I did in such cases, you can use a stropor box to isolate them. So you can keep them in a closed box. You can put some papers that the freezing process is slower, and that was very helpful, and I could use the samples. Using long-term frozen samples, it's more of a facility question. You should have a lot of -80s or liquid nitrogen space, and of course they should also keep stable. I know it is not easy always, so we had also our own issues time to times. When one of the freezer wasn't functioning anymore, you have to transfer the samples and such things. But I haven't had a good experience personally when I freeze them without any isolation. But I don't know, maybe anybody else have experience on that. I had either Mr. Frosty or kind of isolated box that's self-made to slow down the procedure of freezing.
Styrofoam boxes can also work.
Mm-hmm. Yeah.
Thank you very much.
Okay. So I have a question, and it's because of the proper cryopreservation. I saw that in your paper from this year, 2022, the conclusions that you had is 25 years the Buffy coats, and more than 10 years the mononuclear cells, 14 years. Good results. Based with that, how long you think that can be possible to have proper cells cryopreserved, or in future studies you pretend to continue to defrost the cells and try to see how the cells are going on?
I am not 100% sure the difference between the Buffy coat and PBMCs are dependent on the storage time. Or maybe the sample preparation and some other side effects. First, one of the main difference was the Buffy coats from the baseline were in straws. And the second time, they were in small epicaps. And the PBMCs were also in cryotubes. So even the amount and the way of freezing it were so diverse. I'm not sure whether it's the long-term storage or how they prepare the samples. Of course, this prepared also cell density, highly differentiated between the straws and the tubes from the Buffy coats alone. And how they isolated or collected the blood samples in heparin or strut or EDTA tubes were also variously differentiating. So there are so many factors, it's hard to conclude on time point. And I think as soon as you don't culture the cells, you keep them constantly in the same temperature, about -80, I can imagine a longer term preservation would be still possible.
Maybe I can add to that. They in Heidelberg, in the EPIC study, they keep the cells in liquid nitrogen, so it should be possible to use them like forever. I think one of the problems is that in these original samples, we didn't get any samples with like sick people or diabetic people with high damage. So all of the samples were supposedly from normal people, because these are highly valuable samples, and they want to keep the samples, for example, from people who later developed cancer or some other disease. They want to keep these samples for very important purposes, and therefore they asked us to first prove with normal samples whether the method works, and then later we could maybe look for samples where people actually got sick. So from these samples, we don't know if any of them should have had a higher Comet assay damage and we didn't see it. Maybe it works well if we only had some, people had some people in there that were supposed to have a high damage. Maybe we would have found them, but we can't know.
Without further investigation. Yes, I was astonished when you say that in the study that use samples at -60, so -20, it was damage of 10 until 70%. So the temperature is really very, very important.
Yes. I think -60 degrees, that was from older times, and probably most freezers didn't have -80, but instead -60. But even then, for keeping a cell culture seems to be fine. But to me, it seems like the second example that I had, where he said that the doors are always open and people look through the freezers. I think that's something we can relate to. That happens in labs. I mean,
yes.
Yeah, and so I think that's really crucial, really.
Okay. Another question: can we have a Buffy coat after we cryopreserve whole blood, or is it impossible to do that?
Very good question. Never tried it, but then you have to spin them basically down. It should be possible if the erythrocyte doesn't get damaged, but I can imagine if they would get damaged.
Yeah.
Okay.
And then all other cells too. But what we can do in my word, we could isolate leukocytes from also Buffy coat. If we somehow think the Buffy coat is another good matrix for H2O2 treatment, or in general, then it's possible to isolate leukocyte.
And the final one, which you said in your conclusions, and I know that it is a field that you are working on, the Buffy coat field, because if we think that samples in biobanks, the great majority is Buffy coat, the reason why then I ask also if the whole blood we can transform in Buffy coat if we have frozen whole blood. But which more research should be made to optimize the Buffy coat, because we see that there are a lot of factors that are contributing for this. And you even in the paper you talk about the density that you also talk here. So which more research we should make, or even you are doing that to optimize Buffy coat as a good sample to assess DNA damage for the Comet assay?
Difficult question, and maybe I can only comment on it. What we also thought is a second, Buffy coats tend to have a lower DNA damage compared to the PBMCs in that case. And we thought the good correlation might be due to the broader range of PBMC damage, which might be manipulated or played around via changing the electrophoresis duration. So if you have a bigger tail, maybe it's better or easier for evaluating and can give a better correlation. This is what we want to try. And another thing, isolating leukocytes and comparing them whether they are closer to PBMCs is a possible. And of course, in this case, I have the straws and different densities and epis. Of course, this is another ideal case that can be also tested whether it has an effect. I think it had 100% effect during the scoring because of density differences. And when you have 100 samples that you have to hurry on and you cannot leave them longer than, I don't know, we've seen in 2 hours, they have already increased DNA damage. You can't really count all of the cell density from all samples. Yeah, that is still staying as a challenge.
Yeah, I think that was a big issue already, that we could not count all of the samples and therefore had to deal with different cell densities. Now, if we were to plan a new study, we would, of course, adjust the cell density while we freeze them. And also we would include some samples which do have a damage or which we really expect to have a damage, maybe even cells that we have induced the damage, and then freeze them to see whether we can find that. And we would, if we do a new study, actually we're planning to do that, maybe hopefully during this year, a smaller study maybe, but where we would freeze all types of samples, Buffy coat, nicely prepared, and all prepared in the same way, not some straws and some epis at the same cell density, and whole blood and isolated PBMCs, and then compare them in the Comet assay. But that will be a lot of work.
Yes, it will be a lot of work, and very nice work that you are developing together. So thank you again. I saw here that there is no questions. Let me see, there is a raising hand. Yes.
Yes, I have one question. I'm wondering what should be the storage volume in your work? Do you have any suggestion on this subject? For example, 20 microliter or 100 microliter? What do you think?
I think it's for the whole blood and Buffy coats, if you want to store them without cryoprotectant, it is suggested in small volumes, and small volumes mean 200 to 400 microliter, but I work with 200 microliter and was good actually.
Okay, thank you.
Thank you. Thank you for the question. I don't know if someone want to drop. Yes, Gordon.
I have a question. You were talking about generally samples stored at -80. What about samples on well, higher temperatures, let's say on -20? Are they usable for the Comet, or this is too high temperature for storage?
Well, I don't, we have not actually tried, but I think it is too, I think it might be too high. If you look at the literature, then it seems that at -20, the cells already start thawing internally, and it might form little ice crystals, or the tiny ice crystals that are there from freezing form bigger ice crystals and damage the cells already at -20, I think. But we haven't actually tried in a PBMC study or in a human.
But maybe I can tell from the cell cultures, we had somehow some cells that were forgotten in -20 cell lines, and they didn't grow well after thawing compared to the liquid nitrogen. I think -20 is not enough to slow down the whole cell procedure. That's why maybe.
Okay, so -80 would be advisable.
Yes.
Yeah.
Recommended. Yes. So it's cool that errors that we do that are results, research results already. Okay, so some few more seconds if someone can want to drop a quick question. Otherwise, I will do the warm up. Okay. So I want again to show my appreciation to our experts for accepting this invitation and this challenge, because it's the first one, and it was very quick, and they were very responsive to this challenge, and I thank you for that, because otherwise we could not be here, and it was very great in my opinion. So it was perfect that you have accepted. Thank you for all the questions and participation. Hope this was fruitful for all of us. To me, it's sure it was. Any question or doubt that comes to you, you can also email the experts. That papers that we put with the experts also, there are their contacts there. And again, remember that this meeting can be revisited in YouTube, in the ICAWG website. And the second one, Meet the Expert, will be held this year with new topics, probably in October, so be aware of that. And I want to wish you good experiments, great comets, and a very nice weekend. It was a very good way to end this week. It was a very work week for me, so this was the talk. So thank you so much. It was very nice to see you again, and let's see you in a near future. Okay, thank you.
Thank you for organizing this. Have a nice weekend.
Thank
Bye.
you.
Thank you.
Thank you all. Bye.
Thank you very much. Bye-bye.
Thank you.
Bye-bye.
Thank
Ciao.
you all. Nice seeing you all. Bye-bye.
Bye.
Bye.